Problem. While plants do not have a brain or nervous system, they can adapt to multiple situations. However, how well are they able to learn in these different situations? One specific situation we tested was phototropism, which is the plant's response to light. Our hypothesis is if plants were grown at different angles to a light source, then their growth patterns would respond by their stems being angled to the light. For our experimental design, our independent variable was the plant's angle to the light, and our dependent variable was the angle of the plant's stem. The control is the plants directly under the light, and the constants were soil, growing container, seeds, distance from the light, water, humidity, and light source.
The materials you will need for this lab are seeds, soil and growing container, lamp, spray bottle to water the plants, protractor, pencil, and paper to record and measure the angle of growth.
Before even beginning our experiment, we had to brainstorm the most efficient ways for testing our hypothesis. Our first option was to test the intensity of the lights, but with limited resources, we chose to opt out of that. Our second option was to add an angle to the light source, which our team chose to do as seen from the picture included. Now on to procedures. Step number 1, take your seeds or plants and divide them into groups of 2. Step number 2, put a light above the table. Step number 3, take half the plants and put them directly under the light. Step number 4, take the other half and put them at an angle from the light. Make sure it isn't past the light, and also ensure that they are the same distance away from the light. Step number 5, wait until they grow. Step number 6, after they have grown out and their angle does not seem to change, measure them. Lastly, perform chi-square and standard error. Record data and calculate changes.
To measure the angle of the plant, first take your piece of paper and lay it flush with the ground. Second, mark where the base of the leaves are. Then, mark the base of the plant. Now that you have your 2 points, take a protractor or ruler and draw a straight line in between them. Once you have this line, take your protractor and measure the angle. If your line is not long enough, take an extra sheet of paper and extend the line. Make sure that the base of the line is still at a flat end so that the protractor can be used to accurately measure the angle, which right here would be around 125 degrees. Note that this is not an absolutely perfect method and there will be some slight error in your measuring.
Firstly, our table. Our table showed the angle for all plants that survived. If there is a blank, the plant either died or had the seed capsule attached to its leaves, meaning that it could not sense light to its fullest potential. In this table towards the bottom, you will notice a chi-squared value. The critical value for the plants under the light was 32.671 for 21 degrees of freedom. The critical value for plants beside the light was 59.301 for 43 degrees of freedom. The expected value was 90 because it can be predicted that plants will naturally grow perpendicular from the ground. However, if the chi-square value says otherwise, as it exceeds the critical value, it means that there is something else going on that is changing the plant's angle of growth. Now our graphs. In the graph, this shows us the average of the plant's angle, respective to their group. On each bar, there's a standard error bar. This tells us that the data set has a chance to overlap. If they do overlap, it means that the data has no significance as the extremes of the groups could be in the other group also. However, the smaller the error bar, this means that the plants grow at around the same angle, giving the data more significance. Now onto our bell curve. The bell curve tells us the distribution of the plants. This allows us to see not only the mean, but how many outliers were present in the plants. Without the bell curve, we wouldn't be able to see our distribution from all our plants, and it would be harder to see the amount of plants that grew at the same angle. With our bell curve, it is easier to locate extremas and the majority of the plants which grew at the same angles. From the data, it can be clearly seen that there was a correlation between angle of the light towards the plants and the angle at which they grew. Looking at the plants directly under the light, it can be seen that the plants grew as expected. The angle of growth would be expected to be 90 degrees because this is how plants naturally grow in nature because the sun is at a 90-degree angle from them. This was supported in the data because the critical value needed to support it had to be under 32.67 degrees at 21 degrees of freedom. The chi-square value for these plants was 17.04, way under the critical value, meaning the null hypothesis must be accepted and no external forces are influencing the plant's growth. Also, the mean was 92.55, very close to 90 degrees. However, the experimental group, the plants grown at an angle to the light, were extremely off from the expected values, with the mean angle being 142.95 and a chi-squared value of 2479.18. This easily goes past the 67.5 critical value needed for 50 plants. Unfortunately, I could not find the value for chi-square for 44, but seeing that it already exceeds 50 plants, it isn't needed. This clearly shows that there was something extraordinary acting on these plants, given the chi-square value is so far away from the critical point. One standard error is calculated for both groups. It is 1.74 for plants under the light and 1.15 for plants besides the light. There's a huge margin between these two bars, showing how different these two groups were and how similarly the plants grew, respective to their own group. It also means that there is very little chance of the plants being even close to the same angle from different groups. The larger the margin, more difference it shows, which is good. Our bell curve showed the distribution of the plants. In the control, we can clearly see that the majority of the plants were off of 90 degrees by only 5 degrees. However, there are also two massive extremas, around 22 to 23 degrees off. However, that only affected a very small portion of the population. The average difference from 90 was around 55 degrees. In group 2, in the experimental group, the average difference from 90 was around 55 degrees, a huge leap from the 5 degrees difference in the control group. This group did also have extremas, which was around 31 degrees. And even though both groups did have large extremas away from the majority, they still are roughly 10 degrees apart, showing how little similarity these groups have. From the data, it can be concluded that the hypothesis is supported. The hypothesis was the angle to the light would affect the plant's growth, which clearly happened. In our control group, there was nothing that changed the growth of the plant. They followed the expected shown by chi-squared, meaning that on average, there was no change in the plant from the expected values. However, the plants grown beside the light differed from the expected, growing at an extreme angle. The chi-squared value tells us that on average, there was another factor affecting the growth of the plants. This supports that phototrophism is real and had an effect on the plant. The plant groups also had a lot of significance as there was a huge difference between the error bars. This means that the highest and lowest points, each point being a plant, of each mean will not overlap because there's a vast amount of difference between them. The bell curve for each plant also backs up this claim, as not even the extrema from each group did not even get near each other. However, there was one problem with the experiment. The only major problem was that some of the plants ended up dying. Even though this does lower the sample size, the data was still significant, and even though this was a problem, it still had minimal impact on the experiment.
Identify the hormones responsible for the behavior you measured. The hormones that are responsible for the behavior that we measured is auxin, which is responsible for the plant's ability to move, the elongation of cells and shoots, and is involved in regulating plant growth. Another hormone is cytokinin, which was produced by the roots and traveling upward through the xylem. as well as a hormone called gibberellin that regulates growth and influences various developmental processes like stem growth, germination, and flowering.
Post-lab question number 2. Identify the structure and cell type responsible for secreting this hormone and describe how the cells respond to accomplish the behavior you measured. The structure and cell type responsible for secreting this hormone is the auxin. The auxin is a plant hormone produced in the stem tip that promotes cell and stem elongation and inhibits growth of lateral buds. Auxin moves to the darker side of the plant, causing the cells there to grow larger than corresponding cells on the lighter side of the plant. Here is a diagram for visual learners.
Auxin is also a plant hormone produced in the stem tip. It only is produced in the tip and is not produced in the stem, leaves, or roots. However, though, they always are not producing auxin and will only produce it if needed.
This experiment tested how plants grow with light sources being in different locations, one group having the light source above and the other having the light source next to it. The plants in both groups grew towards the light, one growing up and one growing to the side that the light was placed on. The podcast is about Monica Guyano's research experiments, and they show that plants are able to learn. She used experiments similar to Pavlov's dog experiment using the bell and food, but she ran multiple different experiments on the plants, one being that she placed a fan next to the plant so that the wind was coming from the same direction as the plant's light source. And she would turn on the fan and then turn on the light and watch as the plant leaned towards both. And then she would turn them both off, move them, turn on the fan, turn on the light, and then watch the plant move. After a few days, she turned on just the fan and didn't turn on the light, and the plant still leaned towards the fan, expecting the light to come on soon. The podcast connects to this experiment because both deal with the behaviors of plants and how plants are kind of able to learn, because the plants in the experiment grew towards the light, and then the plants in Monica Guyano's experiment leaned towards the fan, expecting the light to come on.
Even though this question may seem unrelated at first, it has a direct relation to this experiment. The first most obvious answer is that without these fungus, plants cannot intake materials such as nitrogen, phosphorus, copper, etc. Without these materials, plants can't create cell walls and cells properly, leading to death or a plant that is extremely limp. In short, without these fungus, no plants would be able to grow, meaning this whole experiment would not be possible. However, there is one material that plants can get without the help of fungi, and that would be carbon. With photosynthesis, the carbon dioxide in the air is converted to sugar, not to be confused with ATP or energy. However, the fungi themselves cannot create their own food or sugar through photosynthesis, meaning they must rely on other organisms to give them food, such as plants. Now the fungi and plant can give each other resources they need if they work together. This leads to the second part of the question. The plants need to grow towards the light, that way they can maximize the rate of photosynthesis, meaning the fungi are also given more food. If the fungi are given more food, that means that they can obtain more minerals and give them to the plants. If the plant's seeds landed in the shade, it would be able to move towards the light, enabling it to start a mutualistic reaction with the fungi and its roots. If plants were static, any plant directly in the shade would have an extremely high chance to die, meaning no new saplings would grow, and the fungus would also die out too. Also, this relationship has to be beneficial. If the fungus were parasitic and leached out the plant would die, especially if not in direct sunlight. However, if the plants only took minerals from the fungus would starve to death also because they can't contain sugars without the plant's help, meaning the plant would then die because it can't obtain nitrogen, phosphorus, copper, etc. This means that plants are trying to get the sunlight as efficiently as possible, which is what the experiment measured. If plants didn't have to give their fungi any sugars, the plants would use as little energy for growing to light because they have no need for being efficient, meaning the angle of growth would be smaller than our experiment showed. Also, assuming the plants give a set percentage of their sugar, which is around 70%, this means that the plants also get a higher quantity of sugar because the total is higher. It also means the fungi can also give the plants more nutrients because now they have the ATP taken from the sugar to do so. Another reason that the plants need the fungus is for production of auxins. Auxins have nitrogen in them. Without them, the plants would not be able to grow towards the light and could only grow straight up. If the fungus stopped supplying nitrogen, this means that the plants would not be able to give back to the fungus, meaning the whole system would die. With the fungus supplying nitrogen, it allows the plants to most efficiently grow towards light, increasing the sugar it creates, benefiting both the plant and the fungus.